Hello, good morning. Today we are going to study about the protein factory of the cell, that is called the ribosomes. Ribosomes are called protein factory of the cell because they perform all the protein synthesis inside the cells, and protein is the main fundamental constituent of the cell, which perform various activities like structural and functional to maintain the vital activities of the cells. All proteins perform various functions. Some protein act as a enzymes, while some protein act as a structural component of the various cell organelles inside the cell. There are many factories, minute factories inside the cells, like ribosomes, which perform various activities to maintain the vital activities of the cells. Today we are going to study in detail about the ribosomes. Ribosome is act like an engine for the protein synthesis inside the cell. Ribosome is a smallest cell organelle inside the cell, and it is membraneless cell organelle inside the cells, which is universally present in both kind of cells, eukaryotic as well as prokaryotic cells. Uh, these minute factories cannot be observed without a microscope. These two protein factories are found in the two forms. First, in the form of free uh, pre living state or bound conditions. So the pre conditions is called in present inside the cytoplasm, and these ribosomes, when binds with the endoplasmic reticulum, they form rough endoplasmic reticulum. The number of uh, ribosomes inside the cell varies. It depends on the metabolic activities or metabolic state of the cells. Those cells which are metabolically more active, they are have more number of ribosomes inside the cell. The number of ribosomes varies from 1000 to 10 lakhs, or we can call 10 millions per cells. Uh, so first we will discuss the anatomy of the ribosomes. Ribosomes are the very minute structures which cannot be observed with the naked eyes, and they are membraneless structures, and they are very minute in size. Their diameter is around 32 to nanometer in size. So the discovery of the ribosomes was observed only after the discovery invention of the electron microscope. If we talk about the structure of these ribosomes, each ribosome is consist of two subunits. The larger one is called larger subunit, and the smallest one is called small subunit of the cells. So we can say ribosome is consist of two unequal subunits. First one is called larger subunit and another one is called smaller subunit. If we discuss the history of the ribosome discovery, the Claude was the first scientist who discovered the who firstly observed the ribosomes inside the cell in the form of microsome as a fraction of the microsome. In 1953, Georges Palade was the first scientist who observed the microscope in individual forms, and he also coined the term ribosomes. So the credit of discovery of ribosome goes Ribosomes are basically present in two forms, ATS and 70S type of ribosomes. ATS type of ribosomes present inside the eukaryotic cells, while 70S type of ribosomes present inside the prokaryotic cells. Here, the S represent the Swedberg coefficient, that is called sedimentation coefficient also, which actually represents the rate of sedimentation during the process of centrifugation. If we compare the two type of ribosomes which are present inside eukaryotic and prokaryotic cells, that is called ATS type of ribosomes are present inside the eukaryotic cells, while 70S type of ribosomes are present inside the uh, prokaryotic cells. If we talk about the location of the ATS type of ribosomes, ATS type of ribosomes present inside the cytosol as well as endoplasmic reticulum, while 70S type of ribosomes present only in cytoplasm of the cell. If we talk about the weight of the ATS type ribosomes, it is around 4 to 5 million Dalton, while 70S type of ribosomes are 3 million Daltons. If we talk about the subunits of the ATS type, the largest subunit of the ATS ribosome is called 60S, and smallest subunit is called 40S type of ribosomal subunits. While in case of 70S type of ribosomes, the largest subunit is called 50S and smaller subunit is called 30S type. Here the question can come, why the 60S and 40S combine together to form ATS. Here this is not a mathematical additions. This is the Swedberg coefficient. On combined, the sedimentation rate depends upon the size or and weight of the constituent molecules. Here the 60S and 40S when combined together they becomes ATS type, that is represent the faster rate of sedimentation as compared to their individual forms. And this is also true in case of 70S type of ribosomes. We can also compare some more details about these type of 70S and 80S type of ribosomes. As we have discussed, the prokaryotic largest subunits of prokaryotic 70S type of ribosomes is 50S, while eukaryotic ribosomes 80S type ribosomes is 60S. A smaller subunit is 30S in case of prokaryotic cells and it is 40S in case of eukaryotic cells. Total number of protein counts which involve in the formation of these ribosomes, that is in prokaryotic cells the total number of around 55 total proteins are present in both subunits, while it is around 70 plus different type of proteins are present in eukaryotic ATS type of ribosomes. If we talk about the constituent of the RNA present inside the, actually RNA is particles or structure which are found by the rRNA, and that is called ribosomal RNA and proteins. So they are basically called ribonucleoproteins. So if we talk about the structural components of the different subunits, the largest subunit of 70S type of RR ribosome is consist of 23S and 5S, while the smaller subunit consists of 16S. While in case of eukaryotic cells, the largest subunit is consist of 28S, 5.S and 5S rRNA, while the smallest subunit is consist of 18S rRNA. And if we talk about the synthesis location of these two subtypes of these subtypes of ribosomes, 70S ribosomes are synthesized inside the cytoplasm, while the eukaryotic cells, that is 80S type of ribosomes, synthesized inside the nucleolus, and while the uh, proteins of these constituents proteins are synthesized inside the cytoplasm, but they migrate to the nucleus, and the assembly of smaller and larger subunits takes place. Normally, during the process of this protein synthesis, these subunits are get assembled only. Uh, when there is no protein synthesis, these subunits are in free state, or we can say they are in the form of their largest and smallest subunits. So first we will talk about the ATS type of ribosomes. Here we can see that the the largest subunit, that is 60S, contains 40 types of proteins and 3 types of rRNA, that is called 5.8, 5.8S, 5S and 28S. The smaller subunits contain 30 types of proteins, 1 type of rRNA, that is 18S. So we can say that this 16S rRNA subunits is a larger subunit, and smaller subunit is called 14S, in combined together to form a ATS type of ribosomes. While in case of 70S type of ribosomes, we can find that there are 50 in case of prokaryotic cells, that is 70S ribosomes. The largest subunit is called the 50S. It is consist of 34 types of proteins and 2 types of rRNA, that is 23S and 5S rRNA. While the smaller subunit of 70S type of ribosomes is consist of 21 types of proteins and 1 type of rRNA, that is called 16S. So these are the basic differences between different type of ribosomes, which we utilize during the process to target the prokaryotic cells while without affecting the eukaryotic cells with the help of antibiotics. The largest subunit of 70S is called 50S, and the smallest subunit is called 30S type of. S here represent the Shadwell coefficient. We will also find that either 70S type of ribosomes or 80S type of ribosomes, there is a common features inside the largest subunit of the ribosomes. Largest subunit of each ribosomes is consist of 3 sites. First site is called decoding site, second is called condensation site, third is called exit site. First, decoding site is also called acceptor site. It is represented by A, while the condensing sites is called peptidyl site, or it is represented with the letter P. Here the peptidyl transferase is an enzyme which formed a covalent peptide bond and during the process of protein synthesis. It is an integral part of the largest subunit of the ribosomes. And third site is called exit site, where the protein get comes out from the after getting synthesized the piece. So we can say that the largest subunit of the ribosome is consists of A side, that is called acceptor side, P side, that is called peptidyl transferase side or peptidyl side, and E, that is represents exit side. So A, P, E are the three sides of the each largest subunits of the ribosomes. So first we will understand the assembly line, how protein get built or synthesized. So first, during this assembly line of the ribosome protein synthesis, the first the um, messenger RNA, which contains the instruction for the protein synthesis, get bind with the smallest subunit of the cell, and it lays down the binding of largest subunits. The subunits of the ribosomes are separately present inside the cytoplasm. When instructions containing messenger RNA, messenger RNA contains the instruction for the synthesis of protein. So when the messenger RNA comes inside, it binds with the largest subunit of the RNA, and with the help of magnesium ion, when the concentration of magnesium ion get increased, then the binding of smaller subunit or these two subunits of the ribosome takes place, and to form a protein synthesis complex. And this complex play important role in the protein cell. Actually, ribosome provides the scaffold for the ordered interaction of all the molecules which involve in the protein synthesis. The other structural components which also play in the process of protein synthesis is called tRNA. tRNAs act as a mediator which brings out the amino acids coded for the particular codons of inside the mRNA, and these tRNAs bring the specific amino acids, binds with the codons with the help of anticodon sites of the tRNA, and uh, the peptide bond formation takes place inside the uh, between the amino acids of acceptor and P site, and this peptide bond formation result in the uh, formation of dipeptide, and this process repeated during the process of protein synthesis, and every time the this larger subunit and smaller subunit get shifted to next codon, and mm, next amino acid is decoded, and this process continued until the whole message on the mRNA get read through this process. We can say that ribosome is a universal cell organelle which is present inside the bacteria as well as in fungi, plants, and animalia. So we can say that it is a universal cell organelle which is present inside each and every type of cells, and it is also ribosomal RNA. It makes the vast component or major RNA. It is around 75% of our total RNA is ribosomal RNA, and other type of RNA are constitute around 25% only. So we can say that these ribosomal RNA are the major components which perform the basic structure of the which are the basic component of the ribosomes, and along with the proteins. So ribosomes are the ribonucleoproteins. We can say that these are the ribonucleoparticles which consist of ribosomes, ribosomal rRNA and proteins. If we if we discuss the ratio between the proteins and ribosomal RNA, in case of eukaryotic cells, the ratio is 1:1. That is, equal amount of RNA and proteins makes up the ribosomes. While in case of prokaryotic cells or 70S type of ribosomes, these are ribosomes are consist of uh, 2 times 2:1. We can say that 2 times are ribosomes and 1 times proteins only. So there is a structural difference and compositional difference between these two types of ribosomes. In spite of this, there are other type of ribosomes are also present inside the some kind of cell organelles like mitochondria and chloroplast. These ribosomes are called 55S type of ribosomes, and the largest subunit of 55S ribosomes is called 35S and 25S. Here we will going to discuss what does the universal nature of ribosomes suggest about life. These we can say that ribosome present inside each and every type of cells. So we can say it is a very important vital component of the cell without which life is not possible inside the cells. So it is a vital protein factory of the cell which is needed by both type of or all type of cells. So it suggests the ancient nature of the ribosomes or RNA. It also suggests that RNA was the first molecule which acted as a genetic material, which was later replaced by DNA. RNA world theory also suggests this uh, notion. So this uh, by this with this we can say that ribosomes are the main protein factory of the cells. Thank you very much.